We're going to talk a little bit more about the cells of the immune system. So I put the ones in here, again, that pop up often on the MCAT. There's a little guide right over here to the left. You can definitely take a read for those on your own time. If you want some more information, you can definitely look it up, obviously. I always encourage you guys to use multiple sources when you're looking at the information. So we're just going to do a brief overview of all of these cells, but I do think they're pretty important. There's not a lot of content, like I said before in this lecture, but I do think the stuff that we talk about is pretty important. So it's definitely worth your while to commit them to memory. And these would be perfect for like flashcards. You can make flashcards. You could use the app Anki for flashcards. You can make, like paper flashcards. I think these are good things to do that with. So the first one we're going to talk about are macrophages. We spoke a little bit about them before. Those are the phagocytic cells of the innate immune system, and they originate from blood monocytes that enter the tissue to differentiate. So monocytes are another immune cell that actually have immune function, and they also have the ability to differentiate. So monocytes will turn into macrophages once they enter the tissue. And they were actually at one point thought to be the same cell, but we now know them to be two different ones. They can also release cytokines in response to a pathogen. And I mentioned cytokines a little bit before, and a cytokine is just a chemical signal released to improve the immune response in a specific area of the body. It's usually like small proteins or substances of that nature. There are a bunch of different ones, but you just really have to know that cytokines are chemicals that are released by immune cells, and they do improve the immune response in a specific area. So they'll recruit other cells over. Maybe they'll initiate an inflammatory response, but a lot of cells do release cytokines to signal other cells. And monocytes, like we just mentioned, they are also another phagocytic white blood cell, and they're the largest of the white blood cells. So monocytes have the potential to differentiate into macrophages and dendritic cells. Dendritic cells are another immune cell. These monocytes are created in the bone marrow. They circulate in the blood usually for 1 to 3 days, and then they enter surrounding tissue to differentiate. So they're doing a lot during their life. In the 1 to 3 days, they're sort of acting as a phagocyte. They're acting as a specific, like an active immune cell. And then in a couple of days, they enter the surrounding tissue and they usually become macrophages, but they will also become dendritic cells as well. That's a picture there to the left. There's you could see the nucleus is sort of split into different lobes. That's pretty common among immune cells, you'll see. So dendritic cells, like we just mentioned, there's a picture of a dendritic cell to the right. It's present in tissue that is in contact with the external environment. So dendritic cells are usually like in your skin, they're in your lungs. I think they are in your esophagus. So those sort of external tissues, the ones that are sort of lining the inside, but they're still considered external, that's where dendritic cells lie. So they also help with immunity right there. So they're derived from monocytes. And when they're activated by a pathogen, they head to the lymph nodes to aid the adaptive immune response. So they're sort of like the lookout. I don't think dendritic cells have too much activity, like sort of destroying pathogens themselves, but they're sort of the lookout. So they head to that lymph node. And the adaptive immune system, they'll usually like T cells or B cells there that will, initiate an adaptive response. So that's why we really consider dendritic cells to be the bridge between innate and adaptive immunity. And dendritic cells can also release cytokines to communicate with other cells as well. So if one dendritic cell, for example, comes into contact with a pathogen, it can release cytokines to activate other dendritic cells around it. So it'll activate other immune cells around it, and it'll just sort of amplify that response. The signal will get back to the lymph nodes. The lymph nodes will probably produce T cells, and the T cells will take it from there. So it really is a concerted effort between the innate and adaptive immune system. So we have neutrophils. Those are also phagocytic white blood cells, but neutrophils are found in the blood. One of the first responders to damage caused by bacteria, environmental exposure, and cancer. So neutrophils are really fast acting. They're usually one of the first ones that are on site. They're usually responsible for like when you get a cut or you get something like that and you see it's healing if it releases pus. A lot of times when there's pus on a wound or something, someone will say that's a sign that it's healing, which is pretty true because the pus is actually dead neutrophils. The pus is created by neutrophils that are pretty much doing their job and trying to basically decontaminate whatever wound there is. And they are the most abundant granulocyte. So a granulocyte is a white blood cell that has granules, and they degranulate is what the process is called, and it releases proteins that are inside of it. So a lot of the proteins have antimicrobial properties. Some of the granulocytes that we're going to talk about release histamine, which is the inflammatory agent that causes inflammation locally when it's released. But neutrophils, they're mostly phagocytic. And if you just keep in mind that they're pretty much the agent that causes pus and healing like pimples or healing bug bites or healing cuts, you should do well to remember neutrophils. Eosinophils are another granulocyte. They make up about 2 to 3% of all white blood cells. And this granulocyte releases multiple cytotoxic proteins. So that's its main method. They usually help combat parasites and some other infections in the body, and they do that by degranulation. So they degranulate, and the cytotoxic proteins will usually kill the parasite or whatever infectious agent is in the body. And they're also associated with allergies and asthma. Eosinophil levels, I know, like from a clinical perspective, whenever someone is being studied for anaphylaxis or any sort of allergy like that, they always check the eosinophil levels because it's an indication of like the patient's condition with allergies and asthma. I'm not too sure specifically about what the indications are, but it definitely is used just to gauge each specific patient's condition in regards to allergies. Okay, and 2 more of these granulocytes are basophils and mast cells, which are usually taught together because they're very similar. They are slightly different. Mast cells and basophils have extremely similar structure and function. Mast cells were once thought to be basophils that had migrated into the surrounding tissues, but are now known to be separate. So kind of similar to the macrophage monocyte thing. They are very similar. They have extremely similar functions, and that's really the main difference. There are other differences like the shapes, the amount of granules that they have, the size of their granules are a little bit different, but their structure and function is basically the same thing, except that mast cells are in the surrounding tissue and basophils are in the bloodstream. So both of them, when they degranulate, they release histamine during degranulation to improve the inflammatory response. So they're responsible for acute and chronic inflammatory diseases like anaphylaxis, asthma, and atopic dermatitis. Again, histamine, anything involving the inflammatory response is going to be involved in any sort of dysfunction like anaphylaxis. Asthma is also an inflammatory disease, and so is atopic dermatitis. So it would make sense that these are the cells that we would look at when we have issues, when we have these kinds of issues. Okay, then we have our natural killer cells over to the left. These are also known as NK cells, and they are cytotoxic lymphocytes. So they destroy intracellular pathogens and tumors. Intracellular meaning inside our cells. So up until now, we've pretty much been talking about like phagocytic cells and cells that degranulate that are basically taking care of these sort of intercellular pathogens. So like bacteria, parasites that don't really need to live in the cell. They could very well be in the bloodstream. They could very well be like in the extracellular matrix. Natural killer cells target intracellular pathogens. So like viruses, for example, they will actually kill the infected cell, like our infected body cell. So they target the infected cell or tumor and they induce lysis or apoptosis. They can induce lysis in a couple of ways. I know one of the ways they do is like they sort of like open aquaporins basically, and they allow water to go in and the cell basically bursts. Apoptosis, obviously we've studied, it has to do with, cascades coming from the mitochondria. It's just cell programmed death. But natural killer cells do have a way to induce apoptosis into those cells. And they do not require an MHC1 proteins to target a cell. We'll talk a little bit about MHC. Those are they just basically present antigen to the immune system. And a lot of cells need that MHC1 complex in order to identify what cells they need to destroy, but natural killer cells actually don't need to do that, which is why studies have recently shown adaptive activity in NK cells. They're being implicated in cancer therapies, That's really where the main focus for natural killer cells and immunology is right now. There's a lot of buzz around natural killer cells and cancer therapy. But we do learn about them in the innate immune system, but they have shown adaptive activity. They formulate antigen-specific immunological memory to aid in fighting against reinfection of the same antigen. So it's not too well known, but they do show some sort of memory. So like if a cell was infected with the same pathogen, a lot of natural killer cells will be able to remember it and recognize it. Okay, so we're moving on to the adaptive immune system. We have the B cells. These cells we spoke a little bit about before. They create antibodies. That's basically what their job is. They are produced and matured in the bone marrow. So you can remember that we have B cells and T cells. B cells, B is for bone marrow because that's where they mature. T cells mature in the thymus. So B, bone, T, thymus. All of them are produced in the bone marrow, but the other ones are, the T cells are matured in the thymus, but B cells are produced and matured in the bone marrow. They present antigens for other immune cells and they release cytokines. So yeah, basically obviously a lot of this function is shared between different cells. And a lot of these cells have multiple functions. So when immature B cells encounter an antigen that fits, they divide and differentiate into plasma cells and memory cells, like we spoke about before. Plasma cells produce and secrete antibodies into the blood. Memory cells have immunoglobulins with the same variable region, but they remain dormant until presented with an antigen a second time. The second response is quick and robust. B cells with the correct sequences are selected and multiplied in a process known as clonal selection. So clonal selection is definitely something you should be familiar with. Basically what it is think, for example, just say we have 1,000 B cells and each of them has a different sequence for a different antigen that they will recognize. If we have a pathogen that's coming in, and let me just define my terms before I continue. Pathogen is anything that is invading the body that will cause an infection or a disease. An antigen is specifically the protein sequence that is on the pathogen. So when I say antigen, I'm referring to specifically what sequence we're recognizing. When I say pathogen, I'm referring to the entire microorganism that is going to cause an infection. So clonal selection, say we have 1,000 different B cells and each of them will recognize a different antigen. When we have this pathogen enter the body, it's only going to react with one of those B cells. And then once that B cell finds the pathogen, it's going to start that one specific B cell is going to start proliferating like crazy and make a bunch of copies of that specific B cell. So that's what we refer to as clonal selection, because the antigen of the foreign invader is selecting the specific B cell, and that specific B cell is cloning so that it creates sort of an army to destroy any other antigens, any other copies of that antigen that may be in the body, or the pathogen that may be in the body. So I hope that makes sense. So moving on to the T cells, we have two. The first of the two we're going to talk about are helper T cells, and these are also known as CD4 cells or CD4 plus cells. They are also written as, and they're part of the adaptive immune system. They have a specific response. And helper T cells are super important. They are also sort of like a surveillance system. Their goal is to activate other cells of the immune system. So basically, whenever they're very, very good at finding foreign invaders. And when they find foreign invaders, they alert B cells and T cells. Because they're not super great at finding them, the T cells are really where it comes from. They release lymphokines and interleukins to aid in immune response. They're cytokines. Lymphokines and interleukins are signaling molecules used to recruit a local immune response. So again, same thing as cytokines. And helper T cells are the target of the HIV virus. So that's what HIV is. It enters your body and it starts destroying your helper T cells so that your body cannot recognize an infection when it takes root. So whenever we're looking at like white blood cell counts or T levels, when we're talking about HIV, we're talking about helper T cells. So obviously nowadays there are a lot of retroviral, antiretroviral therapies. We can keep HIV down to undetectable levels and even positive people. So people who are HIV positive can live very long, healthy lives and not transmit HIV to anyone else. But it's still it's still an epidemic in the world. And I think it might actually be considered a pandemic still. And it's definitely a hot area of research. especially like immunotherapy, specifically regarding the helper T cells. The helper T cells recognize MHC1 and MHC2 proteins. I believe they mostly recognize MHC2 proteins, but have just recently been found to also recognize MHC1. So MHC1, they're they're just proteins that sit on the outside of the cell. So MHC1 are present on all nucleated cells in your entire body. And what they do is they randomly take inner contents of the cell, they chop them up, and they display them on this MHC1 to the outside of the cell. So it's basically just sticking out the internal. So a helper T cell, for example, will come along, check it. If everything is good, if it's a part of our body, then it's just going to move on to the next cell. If it goes to one of these MHC proteins and it sees that there's something foreign, it can understand that this cell is infected, it's got to go. So it'll recruit another immune cell to destroy the cell. And MHC2 are specialized proteins that are found on macrophages and B cells, and they present chopped up antigens recently phagocytosed by the cell. So MHC1 is on all nucleated cells, and it just presents the inner cell's contents at random. MHC2 is specifically for phagocytic cells. They take it in, they chop it up, they digest it, and they knowingly show it to the outside, knowing that it's a virus, knowing that it's a pathogen. So those are the two are a little bit different. So MHC2 is not present on all nucleated cells. MHC1 is. And the other type of T cell are cytotoxic T cells. And they're also known as killer T cells, or CD8 or CD8 plus T cells. And they're just specific cells. They proliferate upon detecting viral proteins on a cell's MHC1 protein. So they'll go around and they'll basically just look at this MHC1 protein that we talked about, how it just chops up at random the inside of the cell and it presents it. So if a cytotoxic T cell sees it, or if it's recruited by a T helper cell, it's going to attach to the cell and it's going to induce lysis and apoptosis, very similar to how natural killer cells do. So it'll either cause the cell to burst, or it's just going to induce the cell to undergo apoptosis as well. Okay. All right, so I'm going to launch the poll again. Take a minute. Which following cells are not phagocytic? Try to answer it without going back to your notes. And again, these polls are anonymous. So take a minute and we will get back to it. Okay, yes. So the correct answer is D, just because all three of the other cells are actually phagocytic. Eosinophils are granulocytes, but they actually do have phagocytic properties. Macrophages are also phagocytic. That's their main function, and monocytes as well can differentiate into macrophages. But before they do that, they are also able to take in and digest other cells, other pathogens. But that's why we're here. We learn. So yes, the answer was D. Basophils are not phagocytic. They degranulate. They release histamine. And they recruit other immune cells to the location, and the histamine also causes a local inflammatory response. So that's their main function.